Hey, my name is Julia Grabow, and I'm going to be talking to you about my research I did this summer. So I was looking at the response of northern white cedars to salinity stress. And it was research that was conducted by me. There you go. So you might be wondering what plant stress is actually. Why should I care about salinity stress or any kind of stress like that? Well, it turns out plants can experience stress in the environments in which they're found. These can be due to heat, just too much sun or too little sun can be really hard on plants. Lack of oxygen can be a problem. They need to respirate. So here's a picture from Ties and Ziegler's plant physiology textbook, and it is showing a picture on the left that shows a plant that has been receiving oxygen at normal levels. On the right is one where it has been receiving too little oxygen, and you can just tell the radical difference in the physiological nature of the plant in those. Water stress is another one. This picture shows, also from Ties and Ziegler's, shows soybeans. This top one shows well-watered soybeans. They're looking good. Mild water stress, not getting quite enough water. The leaves are starting to kind of flip over. And at the bottom, we have severe water stress. That's just a lot of the leaves are just flipped over, and it's just a physiological response to the lack of water. Um, and so, yeah, there are many other kinds of stresses that can happen, but these are just a few examples. And as I've shown, these stresses induce physical changes to the plant. So what exactly is salinity stress, you might be wondering? Well, turns out it is high concentrations of salts in the soil. So you have like ions, like sodium chloride ions, sodium and chloride ions can result in high concentrations of salt in the soil. You can have other kinds of salts as well. It's common in arid and semi-arid environments. So here's a picture of East Central Utah, and it is showing just an environment that is really sparse in plant life. The plants aren't super tall, and so that's just kind of a common characteristic of saline soil or high saline soil environments. Humans can also create saline soils as well. This can be due to agricultural practices, water management practices. There are other things too that can be caused by humans to create higher salinity in soil. And lastly, why is saline soil bad for plants? Well, it turns out that it can lower the osmotic potential of the soil. So essentially, this is just telling you that the plants aren't able to get water as well as they would if the soil wasn't as saline. So it causes a drought essentially for the plants. This can lead to other problems. And I mean, waters need plants, or plants need water. It Saline soils can also inhibit photosynthesis, also a big bad problem for plants. And in bad enough environments or high enough saline soils, toxicity can result in cells or the plant. So there are signs of salt stress, and kind of what I was talking about before. You get stunted growth pretty frequently. You also can see leaves that turn yellow or brown, can be a sign of that physical stress. And here we have a picture from Miyamoto et al. And this is just kind of showing a few different plants in different saline conditions. So on the right of each picture, on the left of each picture, is the lowest saline content in the soil. And then as you go further along to the right, you get more saline content. And you can see here, the plant is looking nice and full, quite beautiful. And here you don't even really have much of a plant. And this is pretty common for most of them. You have stunted growth, not as healthy looking of leaves. So yeah, it's just common in that saline stress. And there are different factors that go into how the plant will respond to the salt damage or the salts in the soil. Plants have different sensitivity levels to salt. Some are very sensitive, others are not. You can think mangroves are not sensitive to salt. They live in salt water. And then you have concentration level of salt in the soil. How much salt is actually in there? As you can see again in this picture, different concentrations in the growing medium results in different stress levels to the plants. And then lastly, how long has the plant been exposed to that salt level? Longer is going to result in more damage. Well, in Minnesota, we don't have arid or semi-arid environments, but we do have de-icing salts, and that has been found to show some increases in soil salinity along roadsides that use that to keep roads clear in the wintertime. So Akiza et al. Found that 50% of roadside sites showed increased soil electrical conductivity levels when they were doing their research. Electrical conductivity is just a method to measure the saline concentration of soil, and it essentially is just looking at how well current of an electrical current passes through water. And here is a diagram from their study, and it's just showing the soil EC levels over several years and of different types of trees. So we have elms, ashes, pines, and spruces. And all of the fourth trees are the ones with the upside down triangles that aren't filled. So here, 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 and here. Those are all in in areas that aren't exposed to de-icing salts. All the other trees are exposed to de-icing salts. And you can see there are significant differences in a fair amount of them compared the de-iced soils compared to the soils that didn't experience that stress. Just showing another visual of what they were finding. How does salt stress actually affect plant physiology in more detail? Well, there actually has been found in Akiza et al. Again, they found that higher sodium concentrations have been found in leaves of plants that grow near roadways. Dmychowski et al. Also found similar findings. This is a graph of the content of sodium in leaves when grown in different urban conditions. So this is the sodium content. These darker gray bars are showing plants that were grown on streets, and then we have urban parks and suburban areas. And this is a category of just damaged trees. But you can tell in the street trees, most of them have a significant difference in sodium levels in the leaves when they're by the street as opposed to other areas. This is the only one, T. euchlora is the only one that doesn't have a significant difference between those. There are two main problems that come about when you have salt in or excess salt in soil. So the salt leaches into the soil, and that is taken up by the root system into the plant, and that can cause problems internally in the plant. But you can also get problems from the salt that sprays from cars when they're driving by, and just the salt spraying onto the leaves and outside parts of the trees that can also be damaging. Specifically in this research, I was interested in northern white cedars, or Thuja occidentalis. This is actually a common landscaping plant in the Minnesota Twin Cities area. It grows natively in more northern Minnesota, which was something I had not realized going into the research project, but was able to find some that weren't as much of landscaping trees. The cedars prefer to grow in areas that are cool, moist, and nutrient rich. They often develop shallow root systems, which I thought was valuable for this research, as the de-icing salts aren't going to, I wasn't sure how deep the salts would penetrate into the soil. So the shallow root system could mean that they might be receiving more salt stress than other trees with deeper roots. Major problems that cedars face: heavy browsing. So deer, as you can see, this cute little deer here, they really like to eat cedars, and that is a really big problem for cedars. Flooding and drought is hard on them, as is wind damage, and de-icing salts are also a problem for them. So it turns out when the salt is in the soil, the cedar has a moderate salt tolerance. It's not super sensitive to salt. It has a low salt tolerance to those sprayed salts, the aerosol salts that land onto their leaves and bark. So here, I'm just going to move my face real quick. Um, Here we have from Foster and Mon. Their study was looking at the salt and how that the salt in soil and how that affects the actual cedars and that they were studying. And so you can see over on this left column, we have different salt concentrations that go up. And then they were looking at sodium chloride and calcium chloride and seeing how those salts might affect the trees. And they looked at the discolored foliage, and they also looked at the roots to see how much damage the roots were sustaining in both. And you can see as the salt level increases, you have a very big jump from the 0.5 molar to the 1.0 molar. Not quite completely significant, but by the time you get to 1.5 concentration, it's significantly discolored foliage. So these cedars are showing problems when they are being exposed to that salt in the soil. And they also found that these saline soils cause cedar leaves to brown, which was part of that discolored foliage they were looking at. And lastly, they noted in their research that the ions seem to build up most in the cedar leaves, actually. So after all of this, I had a few questions. If Minnesota uses de-icing salts in the winter, how does that affect the soils and trees that grow near the roads? Do northern white cedars experience salt stress when they grow near those roadways? And if the northern white cedar experiences salt stress, will it have a higher concentration of ions in its leaves? Kind of just showing that higher concentration of those salts. With all of those questions, my final thought to make this experiment and research more accessible this summer, I wondered if a higher concentrations of ions would result in a higher electrical conductivity level in the leaves of those cedars. So I came up with a hypothesis, which was saying salt stress on cedars will result in a higher electrical conductivity in cedar leaves. My prediction was that if cedars are near roadways, then their leaves will have a higher electrical conductivity compared to those not near roadways. The independent variable is the location of the northern white cedar. Is it near a high traffic area road or a low traffic area, or far away from a high traffic area road? The dependent variable would be the electrical conductivity level of the cedar leaves that I've sampled. So my materials and methods. First, I had to find some collection sites to get those cedars. It turns out that central Minnesota is naturally low in soil salinity. So that made it easy to find places that were far away from roads to find low saline areas. I ended up going to a total of 12 different sites, and you can see here is a map of all of the sites that I went to. These pink reddish ones are the low saline sites, and then the yellow are the high saline sites that I visited. Sample group 1 was the sample group of high saline soil. So these were cedars. The cedars I sampled here were about equal to 30 meters or less than 30 meters away from those major roadways. A good assumption to note here was that I assumed higher traffic would result in more de-icing salt used in the winter. The need to just keep those roads clear would mean that we need to keep those salts on it. I ended up taking 15 samples from 6 different sites of these high saline areas. And here's a map from the Minnesota Department of Transportation. They have a traffic mapping application. This was a sample I took on Round Lake Boulevard, and this application was just really helpful because I could go on here and then look at more information to see how much traffic was actually going on this roadway. Then I had sample group 2. This was the non-saline soil samples. So these samples were collected farther than 30 meters from major roadways. Many were even like dramatically farther than that. Again, I collected 15 samples, and those 15 samples came from 6 different sites. And this is just a picture from the USDA Web Soil Survey, just kind of giving you an idea of the soil salinity levels in Minnesota. So you can see this little mark here is a sample I did in the Cedar Creek area in East Bethel, Minnesota. And you can ignore all of these like little letters going on. The biggest thing is the color. We have a lot of red here. And what that is showing is that there is a 0 salinity soil level for the EC here. So very low salinity. So I had to collect some samples. So when I was doing that, I collected cedars roughly 4 to 5 feet from the ground. There were definitely times when that wasn't possible. In Cedar Creek, the trees were much taller than, the leaves were much higher than that. So I had to reach up and grab those, and some I went a little lower to the ground. When I got a sample, I cut approximately 1 branch that was 15 centimeters, or if I needed a couple branches just due to the size of the sample, I would cut 2 10-centimeter branches. Then I stored those samples in paper bags at a 4°C temperature until I could analyze them. So how did I go about measuring the electrical conductivity of these leaves, you might be wondering? Well, there was a lot of fun stuff to do. First, I got to rinse the leaves in reagent grade water. The purpose of this was to remove any of the salt that might have been sprayed onto them from passing cars. I didn't want those salts to interfere with the actual electrical conductivity readings within the leaves. So I rinsed the leaf samples for about 10 seconds in that reagent grade water. Then I dried the leaves in a drying oven until they were brittle, and that took approximately 24 to 48 hours depending on the sample. I started drying around 30°C, but that was taking a much longer time to get it to that completely dry level. So turned up the temperature to around 60, and I think after 48 hours was the longest any leaf sample stayed in the oven. I then took those dried samples and ground them and measured out 0.15 grams of leaves and then powdered them or ground them with a mortar and pestle. I then added 5 milliliters of boiling water to those dried leaves. And I left the leaves in the water for about 72 hours. So just a little more information on this whole protocol. After those 72 hours, it had turned out that the leaf solution had really thickened. So I added more reagent grade water to it to thin the solution. I ended up having a total of 22 mils of water in the solution. I then tried to strain out larger sample pieces from the solution, not super successfully, but I got some of the bigger ones. I was hoping to not interfere with the EC measurements when taking those measurements. Lastly, I took those EC measurements using the Hanna Instruments 2315 benchtop conductivity meter, as can be seen over here. This cute little machine helped me with my measurements, and I measured all of the leaf samples in microsiemens when doing this. So the results were interesting. First, I want to show you a histogram of all of the leaf EC levels that I sampled. So here you can see the EC in microsiemens, and we have a roughly bell-shaped curve. So that means that the levels are actually pretty normally distributed overall. That's something we want to see. It shows that the samples are kind of following that precedent that a lot of natural things tend to show when measured, that bell-shaped curve. So roughly normal distribution. So then I want to look at those EC levels in histograms with the high saline versus the low saline soils. So this one is a histogram of the leaves EC levels in low salinity soil, or what I labeled as low salinity soil. Again, we have a really nice normality to this. We have maybe one potential outlier here. So it's pretty nice to look at. And just so you know, the mean EC here was 193.3 microsiemens with a standard deviation of 43.5. But then we look at the high salinity soil. This is definitely not normally distributed. We have a lot more uniformity to it. There's no bell shape going on here, and we have another potential outlier here on the right. For this one, the mean EC was 227.2 microsiemens with a standard deviation of 46.4. I just want to note that these standard deviations are really close to each other, which is helpful for future analysis. I want to just show one more visualization of the high versus low saline soil leaf EC levels. We have a box plot here. And so we have the blue down here is the low saline EC values. And you can see just we have the median and the first and third quartile, and then the high saline soils is shown up above. And you can see there is overlap, but it does look like there are some differences going on here. They're not completely overlapping. So that shows there might be a little bit of difference between the two medians of these sample groups. So going back to more of the histograms and the mean that we were talking about, I did want to run some unpaired t-tests. So unpaired because they didn't really have any pairing going on. So I did the unpaired t-tests. The null hypothesis going into this would be that the mean of the low saline and the mean of the high saline soils are the same. There's no difference. The alternative hypothesis would be that there is a difference, that the mean of those soil EC levels are different. So just a reminder, again, we have the mean of the low saline at 193, the high saline at 227. Standard deviation is very close. I did run a few tests of this. So I did a t-test with equal variance, so assuming the standard deviation is the same. And I did a two-tailed distribution and a one-tailed distribution. And for both of these, the p-value is actually under 0.05, which is showing some significance, actually. The one-tailed distribution is 0.02, which is a fair amount lower than the 0.05. I then did an unequal variance, the Welch's t-test, since they aren't exactly the same standard deviation, I just wanted to check that out. And again, it came out as the same results because the standard deviation is so similar on both of them. So it looks like we have a significant difference between the low saline and high saline EC levels of the leaves. I just wanted to give a quick look as well at the variation within each site. So here I have a table that's showing my label of low or high saline content, the site letter, and then the mean of the EC sites and standard deviation, median, and the range. So you can see here, the mean is doing its thing, 133 or 271. So there's a lot of different means for each site. What I would like to draw attention to is the standard deviation and the range of all of these. So standard deviation is really all over for each site. We have 2.1 for our site C, but then you see at site G, it's actually 85. It's a really big difference in the standard deviation. So the sites are really variable between each other. Again, when you look at the median, it's doing its own thing, but when you look at the range, we have a lot of variation. We have B and C have 3, and then we have 120 at G. So it's just all over the place, which kind of makes you wonder about all of the variation within each site. So what are my conclusions from all of this? Well, all of the leaf EC levels are normally distributed. Thumbs up, I like that. The low saline condition was normally distributed, also great. High saline condition was not normally distributed, not as great. The t-test, I'll reject the null hypothesis, saying there appears to be a difference in the EC means between low and high saline soils. So that's saying there might be something behind my whole question I was asking at the beginning. But let's do a double take at this. The t-tests require normal distribution, especially when you have sample sizes that are under 30. My sample sizes were 15, so they're well under 30. when I had the high saline without that normal distribution, that's really calling into question the veracity of the t-test. So I ask, can the p-values actually be trusted with that? It's a good question, I think. The variation between the leaf EC levels between each site varies a lot, and I think that's something worth exploring more as well. I would like to know more of why that's happening. But one other thing, the box plots do suggest some agreement with the t-test. There could be a difference between the leaf EC levels. So I don't think we should throw the t-tests out completely. I think more exploration would be great. So what's next? What is that exploration? Well, non-parametric testing would be great. This is non-parametric testing is just testing when you don't have that normal distribution going on. It isn't quite as detailed of testing as you get with like t-tests and when you can use mean and standard deviation, but it would give us more of an idea of how trustworthy this difference could be between the high and low saline soils. I'd also really like to compare those EC levels with other metrics. When I was doing my analysis of the data, there was potentially some correlation between the overall health of the cedar, how green the cedar was, how much yellowing was going on, and that EC level seemed a little promising. Distance of the cedar from the major roadway and the traffic levels of those roadways compared to the EC levels would also be really interesting to look into. The size of the resin buds actually showed some interesting correlation potential between the EC levels. But wait, why am I talking about resin buds? You might be asking, where did that come from? Well, turns out when I first was starting this research, I wanted to look at how saline soils might affect the cedar resin composition of the cedars. So you can see here, I have this little yellow thing with the two yellow dots on the side. That's the resin bud that is underneath cedar leaves, contains resin. And what I was interested in was looking at the diterpenes and monoterpenes of those resins. So monoterpenes are very aromatic and they often, for cedars, can be used to deter deer from eating them. Diterpenes are another, so these are in the terpene group and they're just like smaller categories and very many chemicals. So the diterpenes are a little less volatile. They don't vaporize quite as easily. So they kind of help those monoterpenes stay around a little longer so they don't just all leave instantly. So I was interested to know how those might be affected by saline conditions. And I did some gas chromatography and mass spectrometry analysis of the leaves. So I found some interesting findings. There were in all of the samples I ran, analyses I ran, there was a noticeable monoterpene in each run, and then there were 1 to 2 diterpenes in each gas chromatography analysis. So here's 2 of them that I ran, and the monoterpene group occurred at the 10-minute point. And so this was a pretty common, this one occurred in all of the analyses, and it varied a lot in height or in abundance, as you can kind of tell here. And then I also was able to find diterpenes that were pretty consistent through each analysis here. And usually there were 2 of them, as you can see here. Sometimes there's just 1, usually this one and more at the 33-minute mark. And so those were pretty common. So it definitely has a lot to look more into. I would love to look into that more as time permits. But yeah, that's where that size of the resin buds comes in, and how does saline soils affect the chemical composition of that resin. I would also like to do more testing. So I'd like to see how much do EC levels within leaves actually vary within one site? Is it common to have a huge variation? Is it odd to have that huge variation? I'd also like to compare soil EC levels with the EC levels of the leaves. Are those correlated? And even more so, I'd like to see what ions are in those leaves and how many there are. And I think that would give a really good idea of whether or not measuring leaf EC levels is a good way to test for salt accumulation in those leaves. So that was everything. I just want to take a moment to acknowledge some really important people who've helped out. Thanks to Maddie and Ethan and TT and Dr. Wilson for their help in carrying out this project. I wouldn't have been able to do this without them. They were really helpful in everything, and it was a great experience to learn more about research doing it. I also want to say thanks to Dr. Jerry Cohen for the use of his lab and of his knowledge. He works at the St. Paul campus of the University of Minnesota, and it was great working in his lab and getting to know his space. Lastly, I want to say thanks to Sasha, who works at Anoka Ramsey County. She gave me a really strong foundation in statistics, and that was really helpful in this. And I also wanted to thank my father, Keith. He helped me apply that knowledge more when I was floundering a little bit when I was looking at all the raw data. So thanks to all of you. I couldn't have done it without you. And that is everything. I just have some more excited here. If you want to look at it later, here you go. And that is everything. Just thank you. And I hope you're having a great day, and I'm going to see how I stop recording. Here we go. Have a good night.